Here's a final lecture about Chapter 18, and we'll talk also a lot about transcription in our class on Thursday. So we're going to keep talking about translation and build a framework for understanding the process that happens at the ribosome, and similarly we're going to build a framework that happens in the nucleus when RNA polymerase transcribes the gene. And here's an image from the book in Chapter 18 showing the DNA. We've looked at this briefly before, and the DNA has organized into different parts and the DNA doesn't change. So the DNA is unchanging, and so when we say insulin, we say the insulin gene is in each of our cells. But it's only expressed, the protein's only made in the pancreas. And how is that possible? And the answer is the promoter regulates insulin so that the insulin gene is only made in the pancreas. But the DNA is unchanging. So the DNA looks the same in all our cells. And how is the insulin regulated? And the answer is that transcription factors, proteins in the nucleus, in the pancreas, bind to the promoter and recruit RNA polymerase to transcribe the gene. So the dynamic part of this story, the difference between the insulin cell and the brain cell, is that transcription factors present in the pancreas bind to the promoter of the insulin gene and activate it. So the promoter DNA is really just like a landing site for these transcription factors to bind and do their job. So the big key kind of story becomes the story of these transcription factors that regulate gene expression. The book tells this story, and the book's story is too complicated. We need to be able to get some take-home messages out of it. And so this is just going to be an attempt to do that. When we're talking about gene expression, here are the three main players. We've got DNA, and the DNA is going to be bound by an enzyme called RNA polymerase. And the RNA polymerase is going to transcribe the DNA template, the transcription unit, and catalyze the synthesis of RNA. So DNA serves as a template for the enzyme RNA polymerase to catalyze the RNA. That's the work that's going to get done during transcription. Ooh, this is a pretty color. All right, so this process is going to be regulated. It's going to be switched on and off. So we inherit all the genes we'll ever need from our parents, and then we switch them on and off in different cells at different times. How does that work? And the answer is that the DNA is organized into these two parts. The part the RNA polymerase does its work on, and to produce the RNA, and the promoter. And the promoter is composed of the distal promoter and the proximal promoter. And these are DNA, so I've just colored the DNA a little bit. So that's still DNA. And then the next thing is, within the DNA, there's a coding region within the transcription unit. That's where the codons are that the ribosome uses in the cytoplasm to synthesize a protein. And the coding region is part of the transcription unit. And the transcription unit is part of the gene. Similarly up here, the promoter has two parts, the proximal promoter, and proximal means nearby. And the purpose of the proximal promoter is it shows RNA polymerase where to land and do its job. And then this distal promoter, and the distal promoter has transcription factor proteins that bind to it that help regulate the RNA polymerase, switching the gene on or off. And so here's a picture of it with everything there. So here's RNA polymerase, and there are multiple copies of it bound to the gene, and it's moving down the gene using the DNA template, the coding DNA template to make the transcription unit into RNA. Then here's a protein that's bound to the partial promoter. There are actually several proteins that bind to assemble this task. And here's a protein that's bound to the distal promoter. And this is just a transcription factor. For example, this could be a testosterone hormone. And this could be the testosterone receptor. So some transcription factors are regulated like that, where they have a binding site for a hormone, and when the hormone binds, boom, they're active, like that. This could be an example of a gene that helps activate new gene expression and new growth during adolescence. And it's been off. Well, this person's boy has been growing up, and now as he grows into a man, the testosterone binds and helps activate these genes that have been silenced and now are active, like that. So that's a cartoon drawing. Here's just another image where I've tried to add something to the book. Here are the transcription factors binding, and the transcription factors are serve kind of like a flashlight, where the gene is off and in the dark, and then when the flashlight binds, it recruits RNA polymerase. The RNA polymerase can see the gene now in the light, and it can activate it. I did that here. Imagine that the promoter has a flashlight bound to it. So this is what RNA polymerase sees. Is RNA polymerase doesn't see any of this DNA that's present unless it gets lit up, and then the RNA polymerase binds and copies it. So the DNA is like words in a book, but it's dark. And the words can't be read until transcription factor proteins bind to the promoter and shine light on the nearby transcription unit. That reasoning, see, that allows us to mix and match. We can use the promoter of one gene and put it in front of another gene, and we can get the flashlight to land and illuminate it. We could take the collagen promoter, and by accident in the skin cancer patient, put the collagen promoter upstream of the PDGF gene. The PDGF gene would be in the dark, but now we have a flashlight bound to the collagen promoter. That's called a transcription factor. And in the skin, there are transcription factors. So this is a skin transcription factor that's going to illuminate whatever collagen is nearby. And in the mutation, the PDGF transcription unit has been translocated and placed near the collagen promoter, and as a result, the PDGF gene gets activated, and RNA polymerase, we can make this a pretty purple RNA polymerase, maybe not. RNA polymerase arrives and transcribes whatever gene is illuminated. And we will stop there and talk about this a lot in class.